So Mercator is actually born 10 kilometers from here in a small village in Flanders. He did his studies in Leuven, which is the oldest university of Belgium. And he actually was the first to coin the word atlas, right? So he proposed this as a compilation of different maps. It's not just any atlas. I mean, you have to see how beautiful this is. It's really with drawings and everything. It's amazing. But it's not just beautiful. He made it practical. So his main point for cartography and what changed cartography forever is that he had this, actually the title is actually super cool. So it's a new and more complete representation of the globe properly adapted for use in navigation. And what he did is that he used the rhumb lines and put them vertical to actually, by a mathematical conversion, allow to have a 2D map of your sphere. And this is still used in navigation today, and it's called the Mercator projection. So yeah, when we started on this Liver Atlas project, and it's funded by the Chan Zuckerberg Initiative, we decided to only submit something when we would have something practical. By that I mean, it's not an atlas if you don't have the real spatial localization of all the cell subsets, but also we really wanted to have flow cytometry markers, to have microscopy markers, so things that you could use to then sort the cells or use clinical frozen samples and so on. We wanted something that would work across species, so in mouse and human, and importantly, a signature and protein markers that would hold in inflammation, so that you would not propose this is this cell, but then from the moment the liver is inflamed, your gene is everywhere and not specific at all. So one last point, this is a very ambitious project. We did it together with Charlotte Scott. So it's 2 labs working side by side to get this done. So we started by CITEseq, right? So it's published in 2017 in 2 fantastic papers. So we decided to get on this. It took us about a year to get this to the maximum. By that, I mean, have the CITEseq so well that it actually works as well as flow cytometry. So we titrated the antibodies, we adapted the staining buffers, we decreased the background, and also worked in a way that we would not use any gene expression profile. So in fact, even with 200 antibodies, we pick up the same number of genes per cell as in the normal 10x system. And so this is the type of results that you get. So the mRNA of TIM4 is on your left, the CITEseq is on the right. So you can see that actually, instead of having this dropout, you now have the protein on all of your cells. In fact, for example, CD14 is much, much better than you have for mRNA. And so for neutrophils, for example, with mRNA, you would not even see the neutrophils. With CITEseq, you can start to see it. And you can see that, for example, they're intermediate for lysic C, very high for lysic G, and it's getting to the level that it's really as good as the flow system. So one last thing I wanted to add before I show you the flow data of this is that just go as broad as you can, right? So CD55, for example, I would never have bought it. It's not in my flow antibody list, but we just decided to make the panel as broad as possible. We did 200. We're now going to try 300 when possible. Again, we see no mortality and no drop in RNA. And the reason you have to go broadly is that it's not because your mRNA is not that great. You know, CD55 would be low, but in fact, the protein is just super good. It means that the clone is very, very good and very, very stable, and that in fact, you can use this in a much better way. So it means that the ranking you had from your normal UMAP or Tisney plot, when you check how much of this is actually available by CITEseq, you already have a shorter list, and how much of that actually really works, it really changes the order of your proteins. And so we then went to analyze this. This is a big shout out for like Nir Yosef's work. Oh my God, like Adam and Zoe, they made this fantastic algorithm. It's called Total VI. So I'm going to show you how good this works. So this is just mRNA, right? So you cluster the same cells only with mRNA. And this is when you cluster it with Total VI, when you have this very good protein signal. You can see that the resolution is much, much nicer. And this comes from the fact that, again, your mRNA, you have a lot of dropout, but if you have good protein markers, it will really make sure that all the cells are clustered together based on the fact that the protein is captured on every single cell. And so this means step 1, the cartography is actually better with proteins. It gives you more crisp heat maps. You can see this is with the protein on the left. I hope you can see my pointer. This is mRNA only on the right. So also the gene signatures will be more clean. But like I said, we really wanted to have this work in flow cytometry. So we loaded the data in FlowJo. So I know it's just 2D representation, but I really, really love this as someone that spent hours and nights on the flow. So this is the UMAP we have. It's of all the CD45 negative cells in the liver. You see it here. You can now load that on FlowJo. You can try any of your 200 markers at any moment, right? So you can really gate on these ones and then zoom in, gate on the other ones. You can try any marker you want until you actually get a whole gating strategy. So just to give you an example, on stromal cells, you use 2 markers here. You zoom in, you say, okay, oh no, wait, that's actually at least 2 subsets on my UMAP. So you look for other markers, you try again. Okay, that's already better, but I still have this sort of tail here. Can I find other markers? And so finally, you can actually make a whole gating strategy in silico to actually try out of the 200 markers, which are the ones that allow me to find all of my cells of interest. And this works on all of the cells in the liver. We can do it for hepatocytes, for all the endothelial cell fractions. So Shalev, this is for you. You can really see, for example, the zonation of your endothelial cells with this, the subsets of fibroblasts, and so on and so forth. So you can now compare them side by side. But one thing I want to go one step further is to which of those will actually work by microscopy. So I'm going to take 2 examples. So CITEseq, which is super nicely zonated on the endothelial cells, that Shalev had that in his paper already, at least at mRNA level. And then 204, which is known as macrophage scavenger receptor 1, which is also zonated, but the other way around. So it's closer to the portal veins. So based on this, I would never have picked 204, right? Because the protein signal is lower. The CITEseq, it was one of the prime markers in Shalev's paper at mRNA level. So I definitely went for CD117, except when we tried this on microscopy, it doesn't work. So of course the mRNA is real. It's just that the protein, it works by flow, but it just doesn't work by microscopy, probably because the epitope is killed by the fixation or something. But 204 work beautifully. You can see the portal area here is stronger, the central area is lower. If you now combine it with live1, which again, the protein is super nice. At mRNA, it's okay, it's not super, but that protein is magnificent. You can see it, all of the endothelial cell in between, but not portal central vein. So now, based on 2 average genes on the mRNA, you can actually distinguish portal endothelial cells, central endothelial cells, your L6 in between, which are in the zone between the portal central veins. You can now do this for flow cytometry, identify all of them with just 2 good markers, and you now have markers that actually work by confocal, so you can directly translate anything you have on the flow on what you have on the confocal microscope. And just for Shalev, you have the zonation here super nicely as well. But so the question is, how do I select those? Because I now have a data set of 200 antibodies by CITEseq, but I want to know which ones will work by confocal. And coming to what Liat has shown you just before, I mean, the MIB is fantastic, but you use 40 markers. Maybe you could use an even better set of 40 markers if out of the 200, you could check which one will work or not spatially. So how do we do that? Well, we decided to go to the Visium platform and try to see, can we get the CITEseq antibodies to work by Visium? So this is the Visium data for mRNA. So we worked together with Marlon and Kartik on this from 10x. So we show them the CITEseq data, and then we try to find an adaptation of the Visium protocol to allow to still have both mRNA and protein detection. So this is the result. You have about 2,000 genes per spot on the mRNA side. So that allows you to find back GLUL and CYP2F2, which again are the super cool zonated hepatocyte markers found by Shalev and Karen in this super nice Nature paper of 2017. And so like this, you can now classify your spots in saying this is portal central vein and all of the zones in between. And now you can compare also your protein data. So for example, CLEC4F is our favorite Kupffer cell marker. mRNA is nice, protein is nice, that's good. But like CD55, I told you before, the mRNA is not that nice, but the protein is actually super nice. It's actually better than CLEC4F. So CD73, mRNA is very weak, but in fact, the protein is super by CITEseq, and the protein also works spatially. And then one last thing, which actually worked by CITEseq, but I thought LY6A and E, I don't even know, is it 2 genes? Oh, forget it. Well, in the end, it's super nicely in the Visium. You can see it's only around the veins, the bigger veins, not in the small L6 in between. So it allows you now to look at mRNA zonation, protein zonation, and so on, and to really start to see which one will work best. And so you had already a ranking due to the CITEseq, but now you can see how much of those actually really work spatially. And then your final list becomes very, very different than your original mRNA list that you had in the beginning. Basically, you now have really practical markers that you can use by flow. and biconfocal. So I'm going to show you a couple of examples of how this works well. So this is done for endothelial cells. So we then went for single cell resolution. So here we use mRNA detection. So what you start from the Visium data. You say, what are my best top 100 mRNA signals that I can really use now spatially to distinguish zones and distinguish the cells. So from that, this is, for example, in the endothelial cell genes, you can see super nice split between your central veins here in pink and the portal veins here in green. So this is using a technology called Resolve that we do in collaboration with Nachiket and Benedict. And in fact, it's done through the VIB Single Cell Accelerator program, which is a very cool system where, as a whole institute, we benchmark single cell technologies. It's just to make sure we can compare them and find the best ones rapidly. So this works really, really well. But for the proteins, we went to Milteni from Maxima. So this is done with Tim and Stefan. So we gave them the top 50 antibodies that we got working by CITEseq and by Visium CITEseq, and we said, could you try this at the single cell resolution? So this is repeated protein stainings, and you can look at it this way. CD38, which is super nice on your L6. You can see it works beautifully by microscopy. This is 105. It's on the central veins, also works. 138, which is supposed to be one of these immune markers, but look, it's superb on the endothelial cells. Is completely zonated from portal to central. And so when you combine all of that, you can start to make a sort of zoom on the different types of cells and their proteins that are very spatially distinct from one vein to the next. You can compare RNA 100-plex on the left with Resolve and then 50-plex with Maxima on the right. And so the other thing that I really like about this type of data sets is now you can start to combine. So for example, we have shown before that Kupffer cells and stellate cells are very close to one another. If you would only have the mRNA, it's well, you see them in pairs like this, but it's not always simple. Some of the spots are distinct. The key here, I think, is that you should always add like general, because you can do 100 anyway. So you should always add general stromal cell and general macrophage markers just to at least find a cell. So if you do that, you will see pairs. But the cool thing about proteins is that you can also now really make a sort of 3D data set with like the whole Kupffer cell in red, which is your macrophage. It's not just next to the stellate cell. You see, it's completely intertwined. They really hold one another across an endothelial cell vessel. So this really gives you additional zooms and additional data that you can now use. So now that you found the best surface markers, you can start to make an atlas, which I think is visually super nice. You can zoom in on the portal area and like this really find the clear bile duct and all of these hepatic arteries and so on that you see here, the lymph vessels here in green. But the last thing that we wanted to do is the following problem. So we have cases where, you now have the 100-plex mRNA and the proteins, but for some of the subsets of cells, you just cannot find only an mRNA or only proteins, because you actually would want a combination of both. The proteins to have the whole body of the cell, and then the mRNA, just because it is the most, the best signal to actually distinguish the subsets. So then we start to work on a system where we would, at a single cell resolution, be able to detect both of them. So for that, we used RNAscope. So it's a system where you can have beautiful RNA signals. But the problem is when you use the normal RNAscope protocol, you're going to just remove all of your protein signal. You can see this, your antigen retrieval basically kills all the F480 MHC2 in this example. So we worked on this to First, you can actually remove the antigen retrieval step. You can see the mRNA is still, it's a bit lower, but it's still okay. The second thing is that you can find your optimal fixation time. So 16 hours is just enough to start to see good mRNA data and without killing the protein. Same for the protease. 25 minutes actually enough to see the mRNA and not to kill too much of your protein. And so altogether, we can now do a panel of, let's say, let me do that again, of 10 markers combined. So it's about 5 proteins together with 5 mRNAs. So you can zoom in, for example, on this zone here in the liver. You can say, okay, those are my 2 favorite markers to split up a dendritic cell from a macrophage. It works across species, it works across tissues. It's MAFB versus FLT3. So you can see this here is a macrophage, and the blue here is a dendritic cell. So that's step one. But now you can actually add protein next to it. And you can see how if I only had the mRNA, this would be my Kupffer cell. Now that I have my protein, you can see actually how long the Kupffer cell is and how it could potentially interact over here with another cell. So now you have mRNA and protein combined. You can do that, for example, for dendritic cells. XCR1 is my favorite marker for CDC1s. It doesn't really work very well, the antibody by confocal, but the mRNA works really, really nicely to find this subset. This here would be a CDC2. Again, MGL2 was not that great by protein, but by gene, it really works very nicely. And so together, we're combining now mRNA and protein to actually make an atlas of all the subsets. Sometimes we combine both and zoom in on each different type of cell in the liver. So it's a massive single cell CITEseq map. We will provide an effective gating strategy that actually works. You can take this directly to the flow, sort the cells, and do functional studies with it. We'll provide which antibodies work spatially and which are the best genes spatially. We'll then provide some maps of single cell resolution, both from mRNA and protein. And in some cases, we'll use both of them where we really need both of them to find the subsets. Now, we're not completely ready yet. All of what I've shown you here is mouse. Of course, we need to do this on the human as well. And one last thing we want to try is also to see, can we look at inflamed settings to see which of these things actually holds. So that's about it. So what I showed you is a mouse atlas. For the liver, I want to thank the clinical people that are helping us to get human samples. So it's Hans van Vlierberger, Lindsay de Visser, Bart van der Borst, and Ruud de Bruyne. So again, this is a massive effort from two teams within the VIB that work very closely together. For the bioinformatics, we work together with Ivan Sais. And then, as I said, the VIB has this super cool single cell accelerator system where we can combine, join forces and really compare side by side all the different technologies. And so many people in that system helped to find the best technologies. And then, of course, the funding for all of this. So thank you very much for your attention. I'll take any question.
Thank you, Martin, for this beautiful talk. I think I'll ask one question of my own, if you don't mind.
Yeah.
You beautifully showed the endothelial zonation. What about immune zonation? Do you see different subsets of Kupffer cells or other immune transcriptional cell states across the lobular axis?
Yeah. So also that is completely structured. It's very interesting to see, for example, that you have dendritic cells mainly around the portal area and not in the other zones. The Kupffer cells themselves, as you know, are zonated. And in fact, it's very interesting, actually. They're closer. The only zones where you have the notch signals and some of the growth factors are the zones where you have the Kupffer cells. So they're not completely across the whole system. They actually have a zonation, and I think it's linked to one of the main niche signals that they need to develop in those areas. So we're looking at this as well. Yeah.
Thanks.
Awesome. So I have a couple questions from the audience, Martin. Kelly asks, how much optimization was required to optimize your CITEseq panel on Visium? And is that a cocktail that's commercially available or is it bespoke?
So the people from 10x are still working on the commercial protocol that would be, foolproof and work all the time. We actually needed a lot of optimization to make sure. It's a bit like what we showed for the RNA scope. If you're not careful, all of the things you do to keep your mRNA actually kills most of your proteins. So, those are the main steps. For example, don't use methanol. It completely kills the protein. So there's a lot of those steps. And the second problem is that you have to be very careful about the background, because otherwise your poly A from the total seq antibody just binds to the poly T to capture the mRNA, and you get massive background all over the place. So yeah, it is actually quite tricky. We've been working on this for a year, to be honest. Yeah.
I can imagine.
Okay, there's another question here from Bingjay Zhang, who asked about, he asked you to explain a little bit how the surface proteins and RNA can be simultaneously captured using Visium. Because when you permeabilize the cell membrane, he's wondering if you won't lose the signals of the surface proteins.
Yeah, so basically, it's very much the trade-off between the fixation permeabilization steps that you do for the protein and not to lose your mRNA too much. But for example, in the Visium kits, they sell these tissue optimization kits just to check if your mRNA is present the thing where the number of minutes you have to process your liver and to see the optimum. For example, the brain is a completely other optimum than the liver to get good mRNA. You can use that kit to check very carefully at what moment do I actually lose my mRNA signal. So we use those to make sure for every single step we kept the mRNA, and then to see that we would get the protein. But in the end, the protein is just an antibody and a barcode, and the mRNA will also catch. They both have poly A tails. So it's not incompatible. It's just making sure your fixation doesn't kill the other signal you get.
Cool. So Jonas had a question about how deeply you need to sequence your protein reads. And he also was wondering whether or not the method worked for giving you more subtle separations between cell type differences. If you were thinking about a healthy atlas versus a disease atlas, where do you sort of draw the lines?
Yeah. The sequencing is actually a very good question. So we sequenced very deeply. We were obsessed with the idea to have, things that would be as good as flow, to really have the best markers. But some people in the Single Cell Accelerator program show that with 10 times less sequencing, you still have enough to find your DE proteins. And so you can actually win a lot of money by not sequencing too deeply. So we went probably way too far. That's a very good point. Yeah, and regarding the inflammation signatures, we don't know yet, to be honest. We hope we'll find some very robust signals, but yeah, that's the most scary part of this project. Yeah, agreed.
Okay, there's another question from C1 Zhang. In the case of discordances between RNA and protein data, how do we tell whether this is a technical issue or true biology?
Yeah, you mean when you have low mRNA, but you catch super nice protein or vice versa? You don't know. I honestly, I have zero idea, but when you have it by CITEseq, in the dropseq system and in the Visium, then I think if that correlation holds on both techniques, I think you're much more secure. But it's a good question. We identified one of the antibodies that really seems to recognize another antigen as well. So it could happen. Yeah.
I guess maybe even isolating the cells and doing some bulk RNA and protein sequencing and seeing whether also there you might have.
That's actually another point. Also on the analysis of all of this, right? Where you have both mRNA and protein, there are very different types of data sets, right? The protein is very good. You catch it most of the time, but there's background. The mRNA, you have dropout. So yeah, we're waiting for, I know, Nir, if you're listening, maybe you can call it spatial VI or something. If someone could find a very good algorithm to combine all of that, that would be super cool. Yeah.
Awesome.
Cool.
Nalini asked a question about what types of samples is this applicable to? Do you need to have a fresh sample? Can you go back and work with archived material? If so, does it have to be, fixed or can it be frozen? What have you found?
That's the scary part. Even things frozen at -80, the proteins at some point start to degrade. So you actually want to work as freshly as possible. I know it's not ideal. Yeah, ideally you would like to be able to pick some clinical data sets from before, or the biobank from your hospital. But yeah, in our hands, the mRNA and the proteins, both of them actually work much better if you use samples as freshly as possible. We haven't tried some other fixation methods and so on. But it's something we should definitely check very carefully to see how many months you can keep it in the freezer and when do you start losing the signal. That's a very good point. Yeah.
Okay, great. Let's take one more question from Ozgun Gokse. Great work. Is it possible to purchase your favorite 200 CITEseq antibodies as a pool?
Well, actually, so first of all those antibodies that we used are sold by BioLegend as TotalSeq A antibodies. So that's the first point. The second is that I think BioLegend is actually working on panels that are pre-diluted, okay, already at the right concentration to use and already completely optimized. So in the future, you'll just buy a panel for cancer, for example, with 200 antibodies completely titrated, ready to go. So they're working on this very hard. Yeah.
Okay, thank you, Martin, for this beautiful talk.